iGEM. We are the Montpellier team from south of France. I know it's a stereotype, but we love drinking good wine and eating good local food. So, are you ready for the next 15 minutes where we speak about oysters? France is in fact the first European producer and consumer of oysters. 10% are produced in the Thau Lagoon, located 30 kilometers southwest of Montpellier. However, oyster farmers are losing each year up to 80% of their production because of oyster infections. These infectious episodes are either due to bacteria, viruses, or algae. They can be harmful for humans or for oysters, both leading to huge economic consequences for oyster farmers. You may be wondering how oyster farmers can save their oysters. Nowadays, they have to rely on specialized companies to perform the test. It is time-consuming and expensive for them. Moreover, while they are waiting the results, pathogens propagate, and in the end, there is no issue to save oysters because it is too late. I'm Emily, and with my colleagues Christelle and Nessim, we will present you the solution of the 2022 iGEM Montpellier team, which is a simple and quick test to detect oyster pathogens. I will tell you about our story. In our way to the encounter with the most damaging pathogens, we did several brainstorming sessions. Meetings with researchers in the field, participation to discussion, and visit to oyster farmers were really important to find the most relevant pathogen to be detected. In the end, we decided to focus on Vibrio asterianus, a bacteria responsible for adult oyster death. To be sure that we were going on the right direction, we conducted a survey dedicated to oyster farmers. We wanted to know if they find it interesting to detect oyster pathogens by themselves and if they think that Vibrio asterianus is a good target to be detected. Our conclusion is that oyster farmers want to detect themselves Vibrio asterianus. This is why our solution is a diagnostic method to detect Vibrio asterianus present in water sample. We want our test to be deployable on the field, accessible for oyster farmers which are non-scientists, and the goal for them is to test water all along the year. So, we have seen that we do not want to propose something that is not useful for oyster farmers. From the beginning, I speak about oyster. I'm not sure that at 10 a.m. it's appetizing, but no, we did not spend our summer to eat oysters. So, how can we detect oyster pathogens? Which technique did we use? Did we manage to obtain results?
Hello everyone, I'm Christelle. I'm going to talk to you about the technique we developed. So I guess you've all heard of the revolutionary technique in biology that is CRISPR-Cas9 because of its genome editing capabilities. Well, we were yet inspired by another enzyme, which is the Cas13 enzyme. And the main difference between the two is that Cas13 can target RNA instead of DNA. So what happens in a basic shell lock reaction? As you see here, we add universal RNA reporters that can be adapted to the detection technique that you want to use. And what happens is, once we have recognition of the guide and the target, the activity of the Cas13 is activated, and so it cuts the target. But also, we have the activation of the collateral activity, which is also another difference between Cas9. It can cut any RNA present in its vicinity. So as you see here, we have cut the reporters. The reporters that are universal and also the collateral activity make this technique highly programmable. And so once we have the reporters cut, we have signal detection that we can quantify. So for our project, we wanted to try two different detection techniques, the lateral flow assay and the fluorescence detection. First of all, we will talk about the lateral flow detection. So what happens is, we take a drop from our sample and from our reagent containing the reporters. What happens is that through capillarity, they will filter through the detection band. And they will bind to one of the two ligands. So here we have two possible scenarios. If the test is negative, we don't have any pathogen that is present, and so the reporters are not cut, and they will only bind to the biotin ligand, with the control band being present. If we have a positive test, which means that the pathogen is in our sample, the reporters will be cleaved and will bind to the antibody, meaning that we have the test band present. And so the first thing we wanted to try was see if we can establish a proof of concept. As you see here, we used a synthetic sequence published by Kellner and al. And as you see on our bands, we have a positive result, which means that our technique can actually detect sequences. As we said before, we want our test to be available for oyster farmers. So we wanted to adapt our experimental conditions for them to use in the field. And as you know, oysters are present in salty water. So we tried to incorporate that in our experiments. We did various dilutions of the tow water in our reagents. And also, we tried to see how the incubation time will affect our test. And from our results, we can say that a 5% presence of tow water in our reagents and a 30-minute incubation period is what's best for the oyster farmers to use in the field. Next, we wanted to see if we can actually detect pathogens. So we did our experiments and saw that we were able to detect virulence genes of Vibrio, which are the TOX-R and the JNA-J gene, which means that our paper-based test actually works. But now, the paper-based test doesn't give us actual quantitative data. So we wanted to try and do fluorescence experiments to have kinetic data to fit to our model. So it's the same principle. We take our sample and our reagent containing our probes, and we put them through the plate reader. As I said before, when the probes are cleaved, they will emit fluorescence, and we can quantify the results using Python pipelines. And so here, the first thing we wanted to see was try to test as many genes as we can. And we were able to do that. We were able to detect the synthetic sequence. We were able to detect Triglyopus, which is a plankton present in the tow water. We were able to detect immunity genes in oysters, muscle genes, and finally, the Vibrio astrianus virulence genes. And so the possibility to detect many genes means that our technique in the future can be multiplexed and easier for the oyster farmers to use. Another thing we wanted to see was how much of the pathogen can we actually detect. And we're able to see that up to 0.01 micromolar concentration, we're able to detect the pathogens, which means that in actual water, this is correlated with the presence of the actual number of bacteria in the water. The last thing we wanted to see was how specific our Cas is. And to do that, we added mutations in one of our sequences, up to 10 mutations in different positions. And as we see here, after 2 mutations, we can't detect anything anymore, which means that it is specific up to 2 mutations. And so finally, we were able to establish a proof of concept. We have a working paper-based test, and also we have a working fluorescence detection assay. And now Nassim will walk you through the model that we developed.
Hi, my name is Nassim, and I'm going to tell you how we modeled the whole system. I'm going to show you sketches of the reaction and how we deduced the model from them. So you start with the Cas, freely diffusing in solution, bound to the guide, and the target also diffusing, and the probes. Then the first step is the association of the whole complex. It's bound by 2 constants, and once this complex is associated, it becomes active. Then you have the unspecific cleavage of the probes, which yield the signal detection that we can measure, and also the specific cleavage of the target, which releases the enzyme, and the cycle starts again. So from these sketches, we're able to draw ODEs and fit the model to our experimental data. So what happens when we try to fit? So as you see from the sketch, the model seems to agree with experimental data, which means we can extract the parameters of the fit and have a deeper understanding on the molecular mechanism of the shell lock reaction. So that's what we did. And we took the mutated sequence and tried to understand a bit more what was happening. So here I'm plotting the first constant, so the association of the complex, as a function of the number of mutation. And as you can see, as you increase the number of mutation, there is a significant drop in this constant, which led us to say that the shell lock reaction seems to be limited by this association rate. And this is also supported by looking at the other 2 constants that we had, so the unspecific cleavage and the specific cleavage, which don't seem to fluctuate as you increase the number of mutations. So to sum up, we have a kinetic model that agrees with experimental data and is able to fit them, and is able to also bring a bigger understanding to the molecular mechanism of our test.
To propose our test to oyster farmers, we have to do further experiments. The first thing is to be able to detect Vibrio astrianus directly. The second is to be able to reach a threshold of 10 nanomolar. For the first, we have to do it in laboratory conditions and then using tow water samples. To reach a threshold of 10 nanomolar, we have to try a simple DNA extraction protocol and the DNA amplification and transcription. So we have seen that we have solutions to further advance our project. But can we have a real impact in society with our Cella company? To answer this big question, we answered the two following questions. Does our product have a place in the current market? And what should we take into account for the development of our startup? To answer that, we followed the classical business plan step. So the first step is to know our environment. We conducted a competing product analysis and a pastel analysis to know the political, economical, social, technological, ecological, and legal events that can have an impact in our society. Our conclusion is that we do not have any direct competitors and we have the place to develop something that it is really for oyster farmers. The second step is to define our product according to the previous step. This is why we conducted the SWOT analysis to know the strengths, the weaknesses, the opportunities, and the threats of our product. Our conclusion is that an added value is to propose service to oyster farmers. Third, we developed a marketing and communication plan. We have found that it is really important to have a user-friendly packaging and to communicate clearly with oyster farmers. Then, we conducted a financial plan according to the product life cycle. We found that it is really important to be present in new markets each year. This is why we have to be innovative and present in worldwide markets each year. But what can we do for the future of Cella? We have to propose service. So first, we can help oyster farmers to the result analysis. And we also think that it is important to propose an app to include Sherlock in participatory science project. So the oyster farmer takes a picture of the test, the result is given to him and to us to implement an infectious model. Then, to go further in the communication with oyster farmers, we did a booklet dedicated to them. It is really important for them to know why they are performing the test and why they have to sample waters all along the year. For the education part, we did a road game dedicated to children for them to understand the basis of synthetic biology and the scientific approach. iGEM was also the occasion to establish collaboration and partnership with other iGEM teams. We also participated to the French meetup organized by Toulouse team, where we won the best presentation and public choice prizes. Our most important partnership was with the iGEM Mohali team, where we found that we have a proof of concept that can be reused for the detection of other pathogens. So here is our big conclusion. You have on the left the negative control and on the right the positive control, where you will see the second band appearing right now. So we completed all the stages of integrated human practices, proof of concept, modeling, perspective, entrepreneurship, and implementation. So our Sherlock project is now closed. So we would like to thank all the people we interacted with and also our PIs, Jérôme, Luca, Christine, Diego, and Guillaume for their help, and also you for your attention.